All right, hello everyone. I want to go through the transformer in single cell omics review and new perspective paper today. I think it's a pretty good paper. Uh, as a review, it's from the famous lab of Fabian Theis, which is located in TUM, and I will try to put most information in this time. So, as you may notice, the paper is about transformers in single cell. What is transformer? It's the famous deep learning architecture with attention head and feed forward networks, which, if you want to know about it more, you can read the paper of Attention is All You Need. And what is single cell? Single cell is a technique to get the transcriptome or other omics data with the single cell resolution. So if you're not from the biology side, I can draw something maybe. So basically the idea is that uh, we have the tissue. Let's say this is a tissue with three different cells. Uh, and each of them has different variety in their gene expression, in metabolites, in proteins they have, and so on. And the question is that how we can study these differences. So basically, the idea is that we put this one cell somewhere and try to get a profile of all genes, let's say G1, G2, and so on, and how much of these genes they have. Or it can be a profile of uh, other omics. So this will be transcriptomics, it can be proteomes, so it will be proteomics, it can be metabolites, it will be metabolomics, and so on. So the idea is pretty simple. The thing is that this uh, variance and this variety between cells, it's not something that easily you can analyze from the data science perspective. And the question is that how we can study such variety better. So that's why we have these deep learning models for different tasks. Uh, so I will, sorry, I will go through the paper and talk about it in more detail. So as I talked about, here we have different single cell data. It can be RNA-seq or it can be a special. So basically additional to the information that you have from each cell, you can have the fact that where are they located in a 2D space. It's information because how the neighbors are uh, reacting can influence your cell and basically your tissue and then your body. Then we have this information, put it through transformer, which has the attention heads and feed forward networks, and then you can have variety of different tasks. You have cell annotation. Basically, it means that you have different cell types in each tissue. For example, in muscle, you have the muscle cells, and you have immune cells and neurons, and so on. And you want to define this label. So basically, it can be a labeling task. But since we have a transformer, we can work with it as a sequence to sequence translation. And then you have uh, your sequence of genes here, gene 1, gene 2, gene 3, and so on. and you translate it to something that uh, sorry, you have cell 1, cell 2, and so on, which each of them has gene 1, gene 2, and so on. And you want to translate it to cell 1 is a neuron, or cell 2 is a this type or that type, and basically you can work with it as a sequence to sequence translation. Then you have cell-cell communication, which basically you want to know how these cells work together, what's the information that passing between them, because we know that lots of uh, biological actions come from the neighboring cells and the signaling between these cells. And then you have cell clustering, which basically it's similar to cell annotation but doesn't have like the cell type, but rather you just want to have some uh, classes or some clusters. Then you have perturbation prediction tasks, which you want to predict what is the gene profile of each of your cells after and before using the drug. So basically you have again your profile, you have your drug, and you want to know how uh, these profiles have been changed. Then you have the gene level tasks. So until now we had the cell level tasks. So basically you want to predict something about the cell, the whole cell, but you can also uh, try to predict things in the gene level. So you have the regulatory networks inference. Basically, you want to know how genes regulate each other. For example, one gene can have some influence on others, or uh, increasing gene A can decrease gene B, or so on. Then you have gene function. Basically, you want to know functionality of the genes, and then the final boss is simulating the whole uh, cell, or have the cell on the silico. So here they talked about the background a bit and preparing data. So past that, go to the second figure. So traditionally, people used to use encoder decoder architecture for such tasks a lot. The cool thing about it is that you have some meaningful latent representation which can uh, be used in some tasks. But firstly, let me explain how you train such model. You have uh, again the gene profile of a cell, then you have an encoder decoder, and try to predict the exact same profile, but with the catch that you will go through a bottleneck that you should pass all maximum information through the some bottleneck, and that force your model to learn some meaningful latent representation. It's cool because if you have, let's say, all right, let's say that I have a tissue including 3 cells. First one is the transcriptomes, which I have some variety. And the second one is the metabolites of same tissue, and it's a spatial omics data. So I want to know how to have a profile for each single cell. So for example, this purple one and the red one should make one virtual cell which contain both red and the purple. And the thing is that like sometimes you don't know exactly how to put this information together, uh, especially if it's non-spatial. You need to have some similarity between these two profiles, and that's not easy task to obtain. So one thing that people do is train an encoder decoder for each profile. So basically try to predict the metabolomics from metabolomics and transcriptomics from transcriptomics. Then here you have two meaningful latent representation, one for each, and then try to minimize the difference between them, and then you will have basically a profile for each cell. So that's one thing that people used to use. And then you have transformers, which can have different training. You have the MLL, or mask language modeling. Basically, you have something like this. You have the gene embedding, and you mask some genes and try to predict them. This is the first unsupervised or uh, self-supervised part, and then you have some task-specific post-training. For example, here we want to predict if it's cancerous or non-cancerous, and so on. Right, then you have your uh, transformer architecture, which we have the multi-attention head, which basically you have your embedding here, multiply it by 3 different randomly initiated weights, try to make 3 different embedding, one as v or values, one as key, and one as query. Then by multiplying the q and k, you try to have your attention score, which basically shows the relevant relatively importance of each embedding token to the other one. So basically, if you have this gene, you will have its importance relative to all other genes, and from this we can have the gene regulatory network, uh, which I'm not 100% sure how much it's accurate, to be honest. Uh, for me, it doesn't make sense that it always has some biological meaning, but yeah, I don't know. Then you have your uh, feed forward network, which is nothing important, nothing special. It's of course important. Just passing this through a network, and you will have your final output. Then you see how much it's equal to what you had in your mind or in your experience, and then calculate the loss, and it will go back this whole way, and you will update all of these weights. Yeah. So then they talked about basic things, and then they have, okay, one important question is that how should we put this information in our transformer architecture. There are 3 main categories. One is by sorting our genes, basically based on how much they are expressed compared to average, or you can have some embedding lookup tables, and you multiply it by your beans, and then that will make your input, or you can have uh, positional encoding and some special tokens. point. Then here you have different training strategy for transformer. You have the uh, MLM, and you have next token prediction, and so nothing important here. And again, you have the applications, which is drug perturbation or general perturbations, data integration, and cell level and gene level tasks that I talked about them earlier. Here you have some famous models from GeneFormer, universal cell embedding, and so on. Usually they are trained on RNA-seq, and usually they are self-supervised learn on the MLM task, but the supervised task can differ between them. And as you can see, you need millions of training data, which is one of the problems with transformers. They are very data hungry, and you can see mostly they train on the encoder only, sometimes on decoder only. And by encoder decoder, I mean this thing that we have here. In A, we have the in mask input prediction, which basically make our encoder, and in B we have our decoder in the transformer architecture. Then we have some other fun things that people are trying, like niche former, which is similar idea, but you have some tokens for the special information. You have usage of LLMs, especially the embedding, uh, using of the embeddings and give it to some LLMs and fine tune it on them. And people had some pretty good experience with that. And it will bring me to the last words of author, which they told us while transformers are cool, we are not sure yet that if we train other models, especially much more simpler models, with same amount of computation, same amount of data, how much, how they will differ and how they will perform. I personally am a bit not positive about it, especially in simpler tasks, and I think it's better to go with simpler models. But of course, nowadays it's hype and everyone wants to train their data with transformers. But we need to wait and see how bioinformatic world will evolve. And with this, I will finish my review on this paper. If you had any question, any comment, if I explained something wrong or vague, just let me know. And if you liked the video, just put a like and subscribe and send it to your friends, family, and have a good day.